And, oh, this always covers up. Okay. Oops, that's not where we're supposed to start. Shoot. Okay. Does everybody see my screen? Okay.
Yep.
Okay. So thanks for the introduction. What I thought I would try to do today is to weave together several stories that we've been involved with over the past several years, looking at the mechanisms that underlie T cell motility. Um, and and thinking about how T cells interpret extracellular cues to to move throughout all the obstacles that they find in the body. So now see, this did this to me the other day. It's not wanting to advance. That's going to be a problem. We should have tested this. I have to. Okay, I am 100% stuck. Hold on. I'm not sure what order of events is going to work better. Let me get out of the presentation. Sorry about this, everybody. Let me try sharing the PowerPoint. This did this to me the other day, and I have no earthly idea why. That works. We good? So,
Yeah.
um, right. So just to start with everybody on the same page, one of the points I want to make is that in order to carry out their role in protective immunity, T cells need to move through a wide range of tissue obstacles. They travel amazingly long distances, and they 1 minute they're moving in a dense 3D environment, then they may find themselves circulating in the bloodstream, moving in 2D along vascular walls, transiting between endothelial cells, and then back again into a dense tissue environment. And so to do all these different functions, they really need to be very plastic in terms of their cytoskeletal behavior, the forces they exert on their environment, in order to navigate all these different obstacles. Oh, now again, we're not going to advance slides. This is evil.
I don't know. Maybe you can use like the mouse like keyboard keys.
Maybe I can to advance with the mouse?
Yeah, or like the right arrow on the keyboard. I feel like that's
No,
good.
that's what I was doing.
Oh,
Um,
okay.
we'll see. We'll see. That worked. So it won't use the arrows and it I don't know. So this is just a couple of movies, this one from Ron Germain's lab and this one from Chris Hunter's lab, to really make the point that in a 3D environment.
Wait, sorry. I
Sorry.
don't see your screen.
Oh no, sorry. All right, that's not working either. I'm so sorry, guys. So one more time. Okay.
Yes, everything's
Yes.
fine.
Great. Well, if I can advance the slide, it is. Yay. Cool.
Yay.
They have to move lots of different ways. We're going to make up time. So really, in order to do this, they have to interpret cues from chemoattractants, adhesive ligands, and the physical nature of the milieu itself to really adopt their migratory mechanisms appropriately. And so that's really the question we're focusing on in the lab today is how do these different environmental cues elicit situationally appropriate migratory behaviors? And if you think about this from a cell biological perspective, it really boils down to two basic processes because we know that in order for cells to migrate, they have to undergo shape changes and generate protrusions. And work from several labs has recently shown that to generate a protrusion, you need two things. You need the one we always think about, which is protrusive force, but you also always need to be able to release cortical actin tethers from the plasma membrane. And these two events need to happen together so that you are able to actually generate a protrusion. So we can think then about how these different extracellular cues control these two cell biological events to allow the cell to undergo shape changes and migrate. So we started thinking about this question several years ago when Nate Roy was a postdoc in my lab, and Nate now has his own lab at SUNY Upstate. Nate came to me early on and he said, you put a T cell down on the integrin ligand ICAM1 and it just goes. It makes an actin-rich lamellipodium and it goes. And I think that's amazing, and I don't think we understand how that works at all. And so that's what he set out to understand. And so Nate began thinking about how adhesive ligands, integrin ligands in particular, allow cells to generate protrusive force. And when we started down this road, we were interacting a lot and still are with the lab of Dan Hammer in Penn Engineering and his postdoc Alex and graduate student Sarah. And Dan's lab really focuses on the leukocyte adhesion cascade, this process through which cells tether and roll and migrate along the endothelium and then pass through. And so Nate and Alex really paired up then and started looking at how T cells interact with integrins that are displayed on the endothelial surface to undergo this 2D migration process. And what they observed right away is that there was a big difference between whether T cells were interacting with ICAM1, the beta 2 integrin ligand for LFA1, or VCAM1, the ligand for the beta 1 integrin VLA4. And you can see immediately that the cell migrating on ICAM1 makes this actin-rich leading edge lamellipodium, and the cell migrating on VCAM1 is really blobby and irregular. These are GFP life act cells. You can see that they do make actin-rich protrusions, but they're short-lived and very different. And so this plays itself out in terms of the amount of F-actin that the cell polymerizes at the interface, and also at how fast these cells can migrate. The cells going on ICAM1 go quite a lot faster than the cells on VCAM1. So here's already a case where two different extracellular stimuli are triggering two different modes of motility. So Nate really was focusing on the ICAM1 behavior, and at the time we were studying this family of proteins called crack adapter proteins. There's two genes, the crack gene and the crack-L gene. As it turns out, in retrospect, everything I'm going to tell you is really a functional consequence of crack-L activity. But Nate was interested in these molecules because he knew they functioned downstream of LFA1 in the signaling cascade involved in outside-in integrin signaling. So we had mice that lacked both of these crack proteins, and Nate and Alex put them on this ICAM1 migration assay. And on the left is a wild type cell, and on the right are two crack knockout cells. And you can see these cells, they actually look a lot like the cells migrating on VCAM1. They fail to make these well-organized actin-rich protrusions. They don't polymerize as much F-actin in the first place. And although they migrate with just as much frequency, they're slower, less directional, and they explore less space. So these crack proteins are somehow needed in the signaling pathway that gets you from LFA1 to actin polymerization and persistent migration. So we wondered then what other kinds of activities this mode of behavior is good for. And one of the things that Nate looked at was migration, transendothelial migration through a blood endothelial barrier. And that is enhanced by treatment of the endothelial cells with inflammatory stimuli. These upregulate the integrin ligands. And if we look at the crack knockout cells, they do that much less well than the wild type cells. And that plays itself out in an in vivo context too. These cells are not nearly as good at getting into inflamed skin in vivo. And then Dan's lab was doing these interesting experiments where they were looking at the ability of cells to migrate upstream under shear flow conditions. And they had shown that T cells and neutrophils can migrate upstream when they're migrating on ICAM1. So the shear flow is coming from the left, and these red tracks are all cells that are migrating upstream. But if you put those cells on VCAM1, they can't do that. And if you take wild type, you take crack knockout cells, even on ICAM1, they don't migrate upstream efficiently. So that then tells us that this uh, mode of migration, which is triggered initially by LFA1 engagement, actin polymerization, allows T cells to migrate persistently, to cross endothelial barriers efficiently, and to migrate upstream under shear flow conditions. And so Dan's lab is continuing work on this, and his student I has been carrying out a screen in this case, a hemopoietic progenitor cell line, looking for other molecules that are needed for upstream migration. And so in these cells too, CRAC-L is needed, but so is Cas-L, which some of you will know is a important force sensing molecule that interacts with CRAC-L, and L-plastin and REM. So these are all molecules that interact with LFA1. But not everything that interacts with LFA1 is needed because if you look at vincula knockout cells, they can migrate upstream just fine. So we're by no means there putting together the signaling cascade that's needed for this response, but we're starting to put together a picture that shows that inside-out signaling from LFA1 to actin polymerization is needed for this mode of motility. And I think the other thing that's important about this mode of motility is all the things it's good for. So 2D migration over long distances, strong directional persistence, passage across blood endothelial barriers, upstream migration under shear flow. So these are all things where a leading edge force is useful for opposing things like shear flow or a barrier that has to be crossed. And I'm not going to go into the data, but we see this mode of motility not only when you stimulate through ICAM1, but also with traditional chemokines like CCL19, 21, CXCL12. And I'll come back to that in a moment. So I mentioned that protrusion is a two-step process, and I've been talking so far about protrusive force. And that's partly because we started this work a long time ago, and that's really what everybody was thinking about then. But now we've been thinking a lot more about this membrane cortex release process and what's involved in that. So, for those of you who don't think about this, the cortical actin cytoskeleton is this dense meshwork of actin that lies just beneath the plasma membrane. And that meshwork is connected to the plasma membrane by a group of linker molecules, most famously and most importantly, proteins of the ezrin, rhodixin, and myosin ERM family. And this gives the cell rigidity and shape, and it also can form a diffusional barrier that can organize specialized domains of the plasma membrane. So we've been looking at these ERM proteins for a long time, and these three proteins, they're highly homologous to one another. They have an N-terminal firm domain that binds to PIP2 in the inner leaflet of the plasma membrane and also to the cytoplasmic tail of cell surface proteins, and then a C-terminal actin binding domain that binds to the actin cytoskeleton. And they can do this linker activity in an on-off fashion, so regulated by PIP2 binding and phosphorylation at this regulatory threonine residue. So if PIP2 is cleaved or the threonine is dephosphorylated, they fall off the membrane. That lets the cell reorganize everything, and then they come back on and lock it down in a new configuration. And we see that with the stimuli I've been talking about. So here is a Western blot for these activated phospho-ERM proteins. You stimulate the cell with CCL19 in this case, and these proteins are dephosphorylated. They fall off the membrane for about 10 minutes, and then they start to come back on at about 15 minutes. And during that time, the cell can undergo major shape changes and start to move. So, how do these proteins factor into signal-dependent T cell migration? So that was the question that a graduate student, Tanner Robertson, tackled when he was in the lab. And Tanner has just taken up a faculty position at Cornell in Ithaca. So he's going to be continuing some of this kind of work, but in zebrafish models. And so what Tanner did was he started by looking at just the distribution of T cells in mice that lack these ERM proteins. And again, it turns out that there's only one of these three molecules that's really important in T cells, and that's myosin. So I'll probably go back and forth showing you data from double knockout mice and myosin single knockouts. It's always the same. So what Tanner noticed, and don't be alarmed by all of this data. There's only two things I want to show you, is that in these knockouts, the T cells accumulate in spleen and thymus. So they are failing to egress appropriately from these lymphoid organs. And in immunology, that is really a hallmark of one chemotactic response, and that's the response to sphingosine 1 phosphate. So S1P is a really interesting chemoattractant. It's a lipid chemoattractant, which is made by the blood endothelial cells and red blood cells. So the levels are high in the periphery, in the bloodstream, and they're kept very low by an enzyme that degrades S1P in the lymphoid organs. So there's always this gradient of S1P that can call cells out into circulation, but the receptor is internalized and downregulated at very low doses of ligand. So as soon as these cells are called out, the receptor is downregulated, the cells are blind to the S1P until they eventually go through the circulatory system, get dumped back into the lymphoid organs, and then they dwell there for a while until they re-express the receptor, become sensitized again, and then they can go out for another round. So this is a really important mediator that directs the duration of time cells dwell in lymphoid organs and calls them back out into circulation. So when Tanner saw a pattern of S1P dependent motility in the Moisin knockout mice, he imagined that these cells might be defective in their chemotactic responses to S1P. And that, in fact, turns out to be the case. So these are transwell assays, and you see the wild type cells migrating out in response to S1P, but the knockouts do that very poorly. And interestingly, these knockouts respond perfectly well to these other chemoattractants. So Tanner found that these cells, again, they have defects in displacement and directional persistence. This is clearly a defective motile response to S1P, and he set about trying to understand how that worked. And he could show that these knockout cells, they have perfectly fine levels of receptor on their surface. And when you give them ligand, they downregulate that receptor perfectly well. That's shown in this plot here. So that's already evidence that the receptor is signaling. And furthermore, if you look at Western blots of signaling intermediates downstream of S1P in the cascade, those molecules are phosphorylated just fine. So the receptor is there, the signals are going through. Why don't the cells migrate? And so Tanner finally got a handle on this when he started just looking at them. And the first thing that he noticed was something about the sort of duration of the response. So these sort of purpley tracks are tracks to two conventional chemokines. And here the cells start migrating when they see the chemokine, and they keep going for 15, 20, 30 minutes. When you stimulate them with S1P, there's this burst of movement, and then it's all over by about 5 minutes after stimulation. So that was already very different. And if we look again at the ERM protein kinetics, we see that with CCL19, these proteins are switched off for a long time, but with S1P, it's only about half a minute or a minute, and then they're back on. So the other thing that Tanner did is to look at the cells when they were actually migrating. And what he saw is that cells responding to conventional chemokines, they make these actin-rich lamellipodia, very similar to what I was showing you from Nate's work on ICAM1. But if you stimulated them with S1P, they do that for a short time, just a couple of minutes, and then they transition to making these membrane blebs. And these are very different from the lamellipodia. They're much more short-lived, just a second or two, and they're fluid filled and just very empty looking. And so if you count the bleb formation, you can see that S1P induces this response, but these conventional chemokines do not. So blood-based motility is not a new thing. It's been around for a long time. And you often will see it with amoeboid cells like dictyostelium or primordial germ cells, but always in the context of confinement. It's something that we think happens when cells are under extracellular pressure and confinement. Then they generate these pressure-based blebs. What's different about what I've been showing you is that with S1P, they're doing this in a complete, like in a dish with no confinement whatsoever. So that means there has to be some sort of cell autonomous pressure being built up in these cells. So to think about what that could be, we did some experiments with Ryan Petrie, whose lab was across the street from ours at Drexel. And Ryan has a technique where he can poke cells with a micropipette filled with fluid and then measure intracellular pressure based on fluid displacement. And he could see that there was indeed increased pressure in the S1P stimulated cells that's not induced by these other chemoattractants. And in thinking about what might be driving that, Tanner thought, well, maybe it's to do with actin-myosin contractility. That's one of the big things that could do it. And in fact, when we stimulate cells with S1P, we get phosphorylation of myosin light chain, which is an indicator of increased actin-myosin contractility. So, so based on that, Tanner looked at the question of whether myosin activity is actually driving this pressurization, and could show that it is because if you stimulate the cells with S1P in the presence of blebbistatin, you don't get this increase in pressure and you don't get the formation of the blebs. So, so that put us sort of here, where you have a chemoattractant signal, in this case, a lipid chemoattractant, that's regulating membrane cortex release and driving increased intracellular pressure as the protrusive force. In contrast, something like CCL19 is doing a similar thing with the actin cortical release, albeit with different kinetics, and driving an actin polymerization based protrusive force. So two different chemoattractants, two different types of protrusive force, and different differential control of this cortical release mechanism. So, but apart from showing you there's different cortical release kinetics, I didn't really explain to you why not having myosin would be a problem for this blood-based motility. And to understand that, what you need to realize is that myosin is one of the key proteins that connects the cortex to the membrane in the first place. So this prevents blebbing, and when a bleb does form, it heals it. The membrane is exposed, like shown here. Here's a cell that's undergoing a bleb. It's got a membrane protrusion with no actin, and then soon thereafter, the actin comes back in. And what's really happening there is that myosin is jumping on the membrane and then binding it back to actin and healing it. So, so Tanner looked at where and when and how blebs form in these myosin knockout T cells as compared to wild type cells. And what he found is in the wild type cells, if you just look in a front to back form and you just score where blebs form, the vast majority of them form at the leading edge or the front portion of the cell. Whereas if you look in the myosin knockout cells, they can't form at the very back, but they can form absolutely anywhere else. And I'm not showing it here, but they're also bigger and longer lived. So basically, in the absence of myosin, you get dysregulated localization of the blebs, and and they're sort of out of control. And if you look where myosin is in these cells, the rationale for all of this becomes very clear. So this is just immunofluorescence staining of T cells with this phospho-ARM antibody that detects the active pool. You see that in the untreated cells, that the ERM proteins are all over the cell membrane. The cell is spherical and not doing much. You hit it with S1P, they're dephosphorylated, they fall off, and then by about 2.5 minutes, and that's right about the time that this bleb response really gets going, we see these ERM proteins coming back on, but now they're coating the rear of the cell. And that creates a weak spot at the front. So when you get an apolar response, like increased pressurization, the blebs are going to preferentially form at the weak spot, and that's going to confer polarity on this whole response and cause the cell to move forward. And if you generate blebs everywhere, the cell can't get any directional movement going at all. So, and then finally, by 5 minutes, when this response is all over, the ERM proteins are back on just like they were at the beginning, and the cell can't move any longer. So, so we think that these ERM proteins, they control the polarity of the response, but they also are responsible for this short-lived duration of the response. Now, exactly why they come back on faster in S1P treated cells versus CCL19 treated cells is something that we're still working on. One reason is probably, and we have some evidence to support this, that the S1P receptor is downregulated much faster, so the active signaling is turned off faster, and that lets the whole system recover more quickly. But I suspect there's more to it. So in this part of the talk, I've just told you that S1P-induced motility is driven by increased intracellular pressure, And we think that this is important for allowing T cells to move through confined spaces and small openings like the ones that they see when they're leaving lymphoid organs. This regulated membrane cortex attachment controls the polarity and the duration of movement, and that's particularly important for this blood-based mode of motility. So, to think about why this is important for blood-based motility in comparison to the lamellipodial mode of motility, um, another postdoc in the lab, Marie Juzin, began looking at the question, and she was looking at what goes on in these myosin deficient cells when you mess up cortical actin linkages, but your protrusive force is driven by actin polymerization. And so I've already told you this mechanism is triggered by ICAM1 and CCL19, among other things. And so Marie started with those stimuli. The first thing she did is she just took wild type and myosin knockout cells and put them down on ICAM1 and had a look. And it was very clear that the myosin knockout cells can make very nice lamellipodial protrusions. Sometimes they're bigger and more robust than the wild type cells, but not always, and that didn't really prove to hold up to careful quantitation. But they certainly can make nice lamellipodial protrusions, and they spread a lot faster than the wild type cells. They're also they move at a faster rate, they cover more distance, and when we looked at these other things I told you about upstream migration, transendothelial migration, all of that works just fine with these cells. So if anything, they're better than the wild type cells in these types of assays. But where things got really interesting is when we put them into 3D collagen gels. So first we looked at what percentage of the cells are moving. And in the collagen gels, I should say, there's no integrin ligand. And this really allows us to isolate this chemokine response. So here's the wild type cells with the percentage of moving cells without CCL19. And then when we add CCL19, so many more cells move. The knockout cells are as good or better than the wild type cells, even when you don't give them any chemoattractant. And the same is true if you look at how fast they go and how far they go. These knockout cells, they don't need a stimulus, they just go. And they're very fast, even without a stimulus. However, they're not more directional. So for those of you who don't think about this stuff every day, there's a couple of different ways to track how far a cell goes. One way is you track the total distance that it traverses, and the other is you track how far it got from its starting point to its end point. And if you divide this crow's fly distance by the total distance covered, you get what we call directionality. If the cell went in an absolute straight line, that number is 1, and if the cell wandered around, it's less than 1. So if we look at wild type cells, they wander more until you give them chemoattractant, and then they make more of a beeline. And if we look at the knockout cells, the same thing is true. So these cells are moving faster and further, but they're not getting anywhere better unless you give them a chemotactic signal. And this other number is forward migration index. It's very similar, except it incorporates a component of directionality towards the stimulus. So that really leaves us in this kind of a situation where we've got this lamellipodial mode of motility. In this mode, the reason the cells can do just fine is because the actin protrusive force has a polarity to it. It's a stochastic polarity, but it's a polarity. So these cells can go if you take away membrane cortex attachment. If anything, you've taken the brakes off the system. So the membrane cortex attachment constrains the basal migration and gives these cells directional persistence when they're migrating randomly. If you give them a chemotactic signal, they're just as good as the wild type cells. And as I've mentioned, this mode of motility is really good for going long distances and crossing certain kinds of challenges like shear flow and junctional barriers. In contrast, the blood-based mode of motility, the forces are intrinsically apolar. So that membrane cortex attachment having polarity of its own is essential for the cell to move forward at all. And we're working on this now, but we think that this mechanism of motility is particularly good for getting through very tight spaces within highly confined environments and across discontinuous junctions that T cells have to pass through as they're going back into lymphatic vessels or, for that matter, out of lymphatic organelles. In the time that's left, I want to switch gears a little bit and tell you a related story that Franklin Stabic Rodriguez, a former graduate student who's now a postdoc in the lab, um, has been working on, which is really trying to understand the signaling pathway downstream of S1P that leads to this blood-based mode of T cell motility. And he's been comparing that to what happens with a better studied chemokine like CCL19 and its receptor CCR7. There we know these molecules stimulate RAC1 that gives you a wave dependent branched actin polymerization that drives lamellipodial motility forward. So what's linking S1P to this blood-based mode of motility? So Franklin, when he started this project, he knew two things based on Tanner's work. He knew that S1P um, signals T cell motility, naive T cell motility, through one receptor, S1P receptor 1. There's 5 receptors, so that's important. Um, and two, I showed you this, S1P activates myosin light chain phosphorylation, and so we know that myosin activity is involved in this process. Now, that sounds good, but it's actually very problematic because S1PR1 is known to signal through the trimeric G protein G alpha I to activate RAC. And myosin light chain phosphorylation typically happens downstream of Rho. So the first thing that Franklin had to figure out, which took several years of his life in retrospect, was how you get from S1PR1 to myosin. Does it involve RAC? Does it involve Rho? And if so, how does that work? So he started with this myosin phosphorylation response because a lot's known about that, and he decided to work up from the bottom. And as I said, there's a lot known about how you get myosin light chain phosphorylation. Canonically, it goes through a pathway involving Rho kinase, myosin light chain phosphatase, as well as phospholipase C and myosin light chain kinase. And usually both of these arms are involved. So Franklin started to look at whether these intermediates are involved. He treated T cells with S1P in the presence of inhibitors, in this case, a Rho kinase inhibitor that totally wiped out even basal myosin light chain phosphorylation. He looked at a phospholipase C inhibitor that inhibits the signal dependent phosphorylation, and he looked at myosin light chain kinase inhibitor, and that had the same effect. So it seems that all of this canonical pathway is involved here. And that really left the big question, okay, does S1P activate Rho? Because it would seem so based on this. So to look at that, he did GTPase pull down assays, stimulating cells with either S1P or CCL19 and looking at Rho activation or RAC activation. And sure enough, if you stimulate cells with S1P, you activate Rho. And you don't really get that if you stimulate with CCL19. Conversely, we knew that CCL19 activates RAC, and when we looked at RAC activation in cells stimulated with S1P, you can see the response is really noisy. But there was no convincing or very little convincing RAC1 activity. And just to try this another way, we looked at phosphorylation of the RAC effector PAC. CCL19 gives really good RAC activation. S1P gives just the faintest little bit of PAC activation, PAC phosphorylation here. So again, little to no RAC activation, but a pretty good Rho activation downstream of S1P. So it looks like this model really is right. CCL19 goes through RAC, S1P goes through Rho, but it still left the question, how does that really work? So we went back and checked and made sure that we hadn't made a mistake. Is it really S1PR1? So we treated with inhibitors. T cell motility is inhibited by S1PR1 inhibitor, but not inhibitors of the other receptors. Um, and Rho activity, and this is important, is inhibited by blocking S1PR1. Okay, so it really is S1PR1. Is it really G alpha I? For that, we treated cells with pertussis toxin, a G alpha I inhibitor, and it inhibited migration and it inhibited Rho activation. So the pathway really must involve S1P to S1PR1 to G alpha I to Rho, and that's a little bit heretical. So, fortunately, round about this time, I was contacted by Henry DeBelli, who was then a postdoc in Orion Weiner's lab and just started up his own lab at UT Southwestern. And Henry was working on this really lovely paper, which just came out in Nature Cell Biology, and I encourage you guys to have a look at it. Um, what Henry was looking at was crosstalk between rack and row, mostly in neutrophils. And he was using a combination of biosensors and optogenetic approaches. And what he found was that you could activate rack, and of course, you get rack activation when you activate rack. But what you also get when you activate rack is row activation at the opposite side of the cell. So that I thought was super cool. And what Henry ultimately could show is that the way that works is that when you activate rack, you get actin polymerization. That increases membrane tension, which is sensed by mTORC2, which then activates row. So there is this, I mean, there's this famous antagonism between rack and row very locally, but it turns out there's a pathway through which rack activates row at long distances through membrane tension as a biophysical intermediate. So Henry was kind enough to help us out in our system using his biosensors. And when we looked with the rack biosensor, we could now detect a transient rack activation downstream of S1P. And it makes sense that we couldn't see this biochemically because this is a single cell assay. You're looking in one place, and it's a very transient response. Here in the blue line is the rack activation downstream of S1P, compared with this sustained rack activation that you get from CCL19. So it does happen. And then if you look at row, there's a bit of a lag. I've tried to line these up in time, and you see the rack activation precedes the row activation, and the row activation is at the side of is at what will become the uropod of the cell, and it's then much more sustained than this rack activation. So this really looked like the relay system that Henry had described in neutrophils. Um, so um, so this is just really the same model I showed you before. We look to see if mTORC2 is playing a role here using an mTORC inhibitor. And what we could see is that this mTORC inhibitor inhibits row activation in the S1P stimulated T cells. And moreover, that inhibitor inhibits myosin light chain phosphorylation. So, so based on all of this, it really looks like the mystery of how you stimulate cells with S1P signal through GFI and get to row involves this tension-based relay system that Henry has identified. So that was very gratifying to see. Okay, so everything was looking good, except that there's one thing I glossed over. Some of you may have caught it. Which is I told you that when we treat cells with myosin pathway inhibitors, ROCK inhibitors, or blebbistatin, we prevent the pressurization, we prevent the blebbing, but I did not tell you we prevent the migration. And I couldn't tell you that because it isn't true. So this was just something that we were completely stuck on and puzzled by for a very long time. If we look at the migration in a transwell assay to S1P, it's not blocked by anything to do with myosin. So we knew we were missing something. And that led us to start thinking about what are some other mechanisms that can control S1P dependent T cell migration. What else could increase intracellular pressure? Well, I already told you it's not extracellular force because these cells are not confined, and it's not myosin. So the only other thing that comes to mind is water influx, hydrostatic pressure. So that was, as soon as we thought about that, that made us think about some work that Victor Tubilowitz had done. And Victor had shown, looking at T cells stimulated with the CCR7 ligand, CCL21, that this kinase Wink1 was important. Wink phosphorylates a couple of other kinases, and these kinases in turn phosphorylate an ion channel that lets ions in and water follows, and this increases the hydrostatic pressure in the cells. And what Victor could show is that that then interferes with membrane cortex attachment and allows the cells to form, in this case, actin-rich protrusions in response to CCR7 signaling. So we wondered, could this Wink1 pathway be involved in the S1P response? And so the first thing that Franklin did is to look at phosphorylation of these two molecules downstream of S1P stimulation. And in fact, both of them are phosphorylated. So it looks like this pathway could be playing a role. So then Franklin went on to do some inhibitor studies to see if you need this for migration. And by this time, we had become a little bit worried about the use of transwells for all of our migration assays. We wanted to do something a little more information rich. And in the interest of time, I'm just going to show you the quantitative data, but Franklin looked not only at migration in transwell assays, but at the formation of blebs and ruffles in response to these stimuli. So if we stimulate cells with S1P, they form some blebs and ruffles in at baseline, but when you stimulate them with S1P, they bleb, but they also form increased numbers of ruffles. And just to make this easier to read, I've put the baseline for ruffles here in red and the baseline for blebs here in blue. If we do this same thing in the response, in the presence of this Wink1 inhibitor, under high dose conditions where we completely block cortical release, there's no migration and there's very, very little shape changes of any kind. These cells just sit there and do absolutely nothing. No blebbing, no ruffling, lower levels than even the wild type cells without a stimulus. So that's good. It told us that Wink1 is involved and it's involved at this cortical release level. But does that relate to the myosin response that I told you about? Well, when we started looking at myosin now, that really was very revealing. So these are cells stimulated with S1P in the presence of the ROCK inhibitor Y27632. And these are the cells that I told you still migrate. Well, now the reason becomes obvious because these cells, it's true they don't bleb, but they make this compensatory increase in ruffling. So you block this pressure-based mode of protrusive force and you actually amplify the actin-based mechanism. So these cells are undergoing this big compensatory change, and that's probably why they can still migrate. But how does that relate to Wink? Well, if we do this, to ask that, we used a lower dose of the Wink inhibitor, one that Victor had shown does not completely block motility. And here we got the same result. We treat with this low dose of the Wink inhibitor, stimulate with S1P. We actually block blebbing, but we don't block ruffling. But we also don't get this compensatory increase. So now we can combine that with the myosin inhibitor, and now we block the stimulation-dependent increase in both blebbing and ruffling, and now these cells can't migrate. So putting all this together, we think that Wink1 is actually playing a role at least two different levels. First of all, it's needed for this cortical release, but second, it's probably working together with myosin to give this increase in intracellular pressure, and somehow it's also preventing this compensatory change that we see with the cells where we just inhibit myosin. So it turns out there was one more little surprise to the Wink1 response because actually in response to a reviewer's suggestion, we looked to see what the Wink inhibitor did to S1P dependent myosin light chain phosphorylation, and it blocked that too. And if you think about it, that makes a lot of sense because I told you that the coupling of S1P to myosin light chain 2 involves this tension-dependent relay system. So if you can't make an actin-rich protrusive response, then you're not going to be able to get myosin activation at all. So actually, this is another level of crosstalk between these two pathways because if some of the signaling is itself force dependent, then you're going to block that if you don't get cortical release. So this is a scary diagram, I know, but it needn't be. So just to put it all together, memory cortex release is achieved by myosin deactivation, mostly probably through phospholipase C cleavage of PIP2 in the plasma membrane, but also by water influx and transient RAC activation downstream of G alpha I increases membrane tension, which in turn activates this mTORC dependent pathway leading to myosin activation. And that myosin contractility, together with the water influx, increases pressure and gives you blood-based motility. But while all that's happening, the cortex is coming back on, becoming reattached again, but now at the rear of the cell, and that biases these protrusive forces towards the front and allows the cell to move forward. So that all contrasts with what happens with CCR7, which is a much simpler situation where G-alpha I activates sustained RAC activation, giving you actin polymerization and a lamellipodial mode of motility. So just to sort of pull this all together, then we're starting to understand different ways that these different extracellular cues control these different motile processes by really regulating coordinately two processes: one, membrane cortex attachment and release. And though we don't completely understand how, we know this can vary in space and time and even degree. And protrusive force. And here we've got two major mechanisms: actin polymerization, which gives a polarized force, and elevated intracellular pressure, which gives an apolar response. And spatiotemporal control of those two steps can give you a lot of different types of motile behavior. And just thinking about this, then it becomes very clear that you have to coordinate these two steps in space and time. So the relay system is one mechanism for crosstalk between them, but there are probably many more that have to allow you to uncouple the actin cortex at the right place at the right time so that your protrusive force can do what it needs to do. So with that, I will just thank the people who did all the work. I went back to the days of Nate Roy in the lab. He's now, as I said, at SUNY Upstate, and Tanner Robertson, who's now at Cornell. So I've now got my gang up the Northeast Corridor. Franklin Stevick Rodriguez is still in the lab for a little while longer. And soon moving on to a postdoc in a colleague's lab, so he'll still be in the family. And Marie Jazan is now in another lab at CHOP as well. And I didn't tell you about the work of Yishen Zhang, but he is pursuing this project now, looking at dendritic cells, where we're definitely finding some things are in common and some things are interesting in very different ways. So that'll probably be the next chapter of the story. And we've had great collaborations with Dan Hammer's lab, Ryan Weiner at UCSF, and Ryan Petrie at Drexel. So with that, I will stop and take any questions.
Wow. Thank you, Jen. That was a really wonderful, amazing integration of physical processes and signaling mechanisms. And I super enjoyed it. Especially when you pointed out that when you finally just started looking at something, it started to make
Yeah,
sense. So
there's
as a
that.
microscopist, that resonates with me a lot.
It took us an embarrassingly long time to just do that.
Yeah, so I see a hand up already. You're welcome to unmute and ask your question. You could also type things in the chat box for me. Oh, hey, Cheng. You can go ahead.
Hi, Jen. Can you hear me?
Yes.
Wonderful talk. Really enjoyed it. I have two questions. Let me start with the first one. So that when you present the ERM protein, the way it interact, it
Mhm.
looks a lot like talin, but you never mentioned that. Um, like the binding of this, I forgot the acronym, this domain to the membrane, and that kind of thing, like the talent would have something like,
Yep.
right? But you never mentioned that, and you didn't say that it's actually, you talk about LFA1,
Yep. 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 Uh, so that's very astute. So first of all, Talon has a firm domain like the RMs, but firm domains are very diverse, kind of like pH domains. You can identify them as related, but they're all over the place. The way I would think about it is the urine proteins have a much shorter attention span than talin. They come on and off very dynamically, even at steady state, if you do FRAP experiments and things like that. And they're really designed, if you will, to be much more regulatable. And to my knowledge, they're not force dependent in the same way that we think of talin and vinculin. So I think there are a lot of interesting differences there in the way these proteins are wired to function. But something that's kind of implicit in your question that I've wondered about for a really long time is all of these sort of focal adhesion domain or focal adhesion proteins, especially the ones in that sort of first layer, are how are they contributing to this kind of connectivity? And in the when we've looked, they're mostly not. So, or at least in a very fundamentally different way. T cells, we did some work with pattern surfaces and stuff. We could force a T cell to make something that you could see as a collection of focal adhesion proteins. So they do utilize all of them, but they make very low adhesive contacts. In fact, when I talk with people that you will know, Paul Janmi and people like that, they're like, oh, these cells, they don't exert any force to speak of at all. They're not even interesting. So um, it's all much lighter touch and much more dynamic. But those guys, they have to be playing a role too.
So I want to follow up before I move to the next question. I thought the T cell receptor also connects to cytoskeleton somewhat through the IM protein. I know the data was not definitive, but I remember seeing people saying that.
I have seen people say that. I don't think it's right. I
Okay.
there is a story about the T cell receptor complex through NCK interacting with the cytoskeleton as well, and I think that is true. But every time we look without integrin, it's pretty negligible.
But you put LCK in the slide where you put a TCR there, you get this.
Because you do need SERT kinases in this cascade. It's all in there. It's all in there, but I do think it's very contextually different.
All right, so my second question, real quick. So you had two mechanisms for migration, right? One is like the protrusive force
Yeah.
through actin polymerization. The other one is intracellular in pressure, right? So
Yep.
I thought that you said when this ERM protein kind of transient release and it created a weaker spot. So
Yep.
you don't have to increase intracellular pressure. You just have to create a weaker spot so then they would get bulged out. But if you say that there were water influx, then you could have increased pressure. Do you have any measurements to say that the intracellular pressure was increased?
The only measurements that we have are the ones that we did with Ryan. And there it was very clear that you do increase the pressure when you stimulate with S1P, and you do block that if you inhibit myosin. We haven't gone back now to look at the aquaporins and to see to what extent the water increases the pressure. Um, and
Well, another mechanism that you can increase the cellular pressure was the contractile of the actin, right? So you have the volume liquid inside, creating pressure. You contract to squeeze it, then you create pressure in a directional way.
Exactly. And so I mean we do have those direct measurements that pressure does go up. The relative contributions of myosin versus water influx, we don't really have. And I suspect the water influx is less because we can pretty much zero out the pressure increase if we just inhibit myosin. So and I don't want to leave anybody with the impression, I might have done that, I didn't mean to, that you don't make a weak spot if you need an actin protrusive force. You absolutely do. So the ERM proteins go to the back of the cell in that case too. It's just that the cell doesn't need that in order to decide how to go forward. Whereas I think with blood-based protrusions, it absolutely needs that.
Thank you.
Sure.
Brent?
Yep. It's always hard to follow Cheng. So I think you kind of hit on one of my questions is that you had a lot of the slides. That was not a complaint, that was a compliment, by the way. That you hit on my question I was going to ask, kind of if you thought some of the release of the Moisin, I guess, or ERM in general could be force sensitive. And so are you kind of, because Alex Dunn does have this kind of uh, paper that Ezrin will form a slip-on with actin, and it's also a little bit loaded. I'll admit we have an Ezrin tension sensor, not Moisin, unfortunately, but Ezrin that does bear some load. So I was just kind of wondering your thoughts about chemical versus maybe force-based
Yeah,
regulation of the release.
super interesting question. And I pitched this. So my contiguous lab mate is Guilherme Nader, who came from microfluidics and dynamic compression world. And so one of the first things I pitched to him when his lab moved in is, let's squish some T cells and see what happens to the RM proteins. And somehow or other, it never got done. I like you, I think, bet that there will be a pressure sensitive release, and I think that would be super, super interesting. But then the devil will be in the details. Is it because the pressure induces lipid cleavage? Is there actually a signal cascade there as well? And
Yeah.
I think that will prove to be the case. And until we start integrating this biochemistry and biophysics in the same experiment, we're not going to be looking under the right rocks.
Mhm. Well, cool. Thanks for a wonderful talk and a great answer.
Maybe I can sneak in one last question. So I think a lot about endocytosis,
Hmm.
and that also can be regulated by membrane tension and is also regulated in cell migration. And
Yeah.
so I wonder if, you've thought about that at all, or what your thoughts are on if that could be like another process that's being heavily regulated by this really interesting like tension crosstalk, where you also kind of want to increase endocytosis at the rear of the cell and decrease it at the leading edge, where you're kind of taking up receptors and then maybe delivering them to where the cell is going to.
Yeah. I mean, we've definitely thought about it. I don't know that we've thought about it well. It starts to make my head hurt a little bit because the receptors in this case themselves are regulated by endocytosis.
Mhm.
So the signal duration may be very dependent, certainly with S1PR1 on endocytosis. And then you have the potential for some of these receptors to signal through endocytic pools and others not. So the experiments are going to have to be designed to deconvolve all of that. Um, but once again, I think you're absolutely right. I think that that will be part of the thing that we need to figure out. And it might be that whether that the membrane tension that's exerted is itself affecting the rate of receptor downregulation, which so there can be a
Yeah.
feedback, right? So yeah, it's a super interesting question. We've done the only thing we've done that's even close, and it's not directionally the way you're thinking about it is because we know that S1PR1 is internalized in a fashion that involves Greek 2. Then we've treated T cells with Greek 2 inhibitors and shown that we can extend the duration of the S1P signal, not as long as CCL19. So maybe that's not the whole answer, but definitely one of the things that makes for that short response, and we don't know yet about the ERM proteins in that, is the endocytosis of that receptor. And in dendritic cells, it's a different receptor that's not internalized, and the game is different.
Yeah. Okay, cool. Of course, it just more layers of complexity.
Yeah, it'll keep us all in work.
All right, great. Well, I think we hit 12 o'clock, so let's thank Jan again, and I guess we'll see everybody again next month. Thanks,
Okay,
everyone.
thanks everybody.
Thank you so much, Jan. That was really
I hope it wasn't too much too fast.
no, not at all. And I'm not an immune cell person, in case you couldn't tell, but
There's a
um,
lot of fear of immune cells out there. We try to counteract that. And
no,
just
you
think
did a great job because I didn't get
just
confused at
cells.
all. Um,
Well,
yeah,
thank
no, but I think the idea we've been doing a lot of stuff like modulating actin as well as like extracellular pressure and looking at clathrin-mediated endocytosis. And the two pathways are definitely like intersect even in like the fibroblast cells that we look at. But
Yeah,
yeah,
the other person
like,
that we've been talking to a lot is Ben Weiner, who was also in Orion Weiner's lab. Um,
mhm,
and he thinks about phagocytosis and membrane reservoirs and things like that. So,
yeah,
so that's also going to be part of this membrane tension and all of that kind of stuff. So yeah, and we worked for a while on a protein called HS1, which is the hematopoietic homolog of cortactin,
mhm,
and is very important, I think, when you need a little bit of extra force generation in your endocytic pathways. We've never gone back to HS1 to see if it plays a role in this stuff. That'd be interesting.
yeah, that would be cool. Great. Well, thank you again so much for
Thank
being
you
here
guys.
and
me. It was
Yeah.
fun.